My path was a little bit all over the place. I took a year off and worked in industry just to take some time off, a little bit burned out after my undergrad. And that was the year that the human genome was sequenced. And I kind of naively thought, well, I guess we know everything about the human genome, so maybe I need to study all the proteins that this genome encodes. And at the time, proteomics wasn't a field. It was just a thought of, or a concept that maybe we could analyze very complex biological mixtures of proteins using a technique called mass spectrometry. And that's what attracted me to the graduate program that I entered to learn how to use mass spectrometry to sequence proteins. And there I met a researcher that was very interested in chromatin biology named David Alice. And we started collaborating with him. And I started getting really attracted to this field, which at the time was just known as transcription and chromatin biology, but soon would become the field of epigenetics. And fast forward a few years later, a little bit more training, and then I was kind of caught in between two fields that were really emerging, proteomics and epigenetics.
So I actually wanted to rotate in the Garcia Lab because when I was an undergrad, I collaborated with the lab and I got to know some of the people in the lab, and it seemed just like a really fun atmosphere. I wanted to learn more about mass spec and proteomics, and I've always wanted to do research with epigenetics, so I thought this would be a good place to try out.
I think something that I really enjoy is learning all the new technology coming up. I like the fact that it's fast and gives us a lot of reliable data, and how we can quantify how these modifications are actually affecting your epigenome.
So what I love about mass spectrometry is its ability to identify and quantify proteins, and it's a technique that's very underutilized in pediatric cancer. And how I want to develop as a scientist is by using different approaches. It can help us find the best targets to then generate the best therapies. So that's one of the big reasons I love mass spec.
So we are a group which is growing, and we have so many instruments, as you can see: classic, triple Q. There's a lemon which is actually a QE, HFXs, baby fusion, daddy fusion. So we actually need a scheme where we can make sure everybody gets time to run their samples, and so I think this is actually the grease that keeps the machine running smoothly, I would say.